Hello everyone and Happy New Year. And today we're gonna talk about the recombinant DNA. So first we're gonna explain what is the recombinant DNA and explain the steps and the aims of the recombinant DNA. So let's start with the definition of the recombinant DNA. A recombinant DNA is a technology or is a technique in which a fragment of DNA from a donor cell or an organism is isolated and then inserted into the DNA of another cell or organism, and this allows scientists to introduce a new characteristic into organism by inserting a new DNA into it. As you can see in this diagram, there is some steps that we need to consider and to add a DNA to a plasmid vector, so it could have a certain shape and certain characteristics. So now we're going to talk about the steps of the recombinant DNA, and first we have to get the bacterium cell and extract from it the plasmid, and a plasmid is a small circular double-stranded DNA molecule that act like vectors to transfer the gene of interest into the host cell. And now we get the cell that contains the gene of interest and get the DNA fragment containing the gene of interest that is obtained from the donor cell, so it could be transmitted or added into the host. And after obtaining the plasmid and the DNA strand or the gene of interest, we're gonna go deeper inside the plasmid and also inside the gene of interest. So we could find there the new enzyme that cut the plasmid and also the DNA itself into a shape that fit both the DNA and the plasmid into each other. And this enzyme is called the re-restriction enzyme. This enzyme cut the DNA into a special or a certain base sequence, leaving a sticky ends in both the plasmid and the gene of interest, so they could fit together. When both the plasmid and the DNA fragment are both cut into a certain shape by the restriction enzyme, here comes the job of the ligase into the fourth and into the last step of the recombinant DNA. A ligase is an enzyme that acts like a glue, and it's used to join the two pieces so they could form the new recombinant DNA. And after making the recombinant DNA, go to another step, which is the insertion of the recombinant DNA into the host cell. When the plasmid is finally inserted into the host cell, the DNA replicates like any other cell, leaving the same plasmid, and the daughter cell will remain genetically modified. And now we're going to talk about the aims and the application of the recombinant DNA. And first we're going to talk about the human insulin. So scientists built the human insulin in the lab, then they remove a loop of bacterial DNA known as the plasmid, as we have said before in the beginning of the presentation. And after they do this, they insert the human insulin gene into the plasmid. Then after they insert it, it will be genetically modified. And after that, the researchers return the plasmid into the bacteria. So they put the recombinant bacteria into a large fermentation tanks, and fermentation is the metabolic process that produces chemical changes in the organic substrate through the action of enzymes. And in this fermentation tanks, the bacteria replicate, in which the human insulin will also replicate, and then it get extracted as we see at the next slide. The recombinant bacteria use the gene to begin producing the human insulin, and after that scientists harvest the insulin from the bacteria, and they purify the substance for the use of medicine for people. And finally, we're going to talk about the use of the recombinant DNA in food industry. And in food industry, use various enzymes that are generated with the help of the recombinant DNA technology. Enzymes produced by the recombinant DNA technology are used in food production and food processing, in which this technology is widely used in food industry to produce different food products, such as the genetically modified food to enhance the taste, to enhance the yield, and to increase the shelf life and to improve the nutritive value.